Yeah, hi everyone. Thanks for being here. Thank you for inviting me. It's a great opportunity to be able to talk to this expert group of people about my work. This is actually the work that I had done during my PhD at CU Boulder in collaboration with my PhD advisor, Dr. Orit Peleg. This is one of my projects there, but I do want to, like, if I get time in the end, I do want to just like give a general overview of what I'm interested in, which the theme is really very similar. And I really had a hard time choosing a project for this talk because a lot of my projects could sort of be related to this audience. So I hope you enjoy this one. It's about adaptive honeycomb architecture, mostly after I heard Professor Feinerman's talk about ant architecture, I kind of wanted to sort of make this the center of my talk so we can maybe discuss and figure out like distinctions between what we call embodied cognition, self-organization in biology, physics, and so on. So I hope there will be nice discussion about it. So before starting, just like going straight into honeybees, as I referenced the work on the weaver ants, I think there are other ants who make nests that are orders of magnitude larger than the individuals. And then there are honeybees that build nests like that, and also termites. In all of these examples, there are no these individuals are limited. There are no like extraordinary ability in one individual. There is no direct access to what the global picture or the large picture in the end will look like. And there is no central blueprint or leader in any of these processes. So really the nest structure I think in all of these examples is really the emergent property of the collective effort. And I do want to focus on the honeybee nests, and I hope I can sort of convince you with the controlled experiments that we did that there is, so the behavior or the structure at least, if not the behavior, is a combination is a combination of the individuals, their physical bodies, and their environment, like how the environment affects it, which is kind of the theme of the talk. So honeycomb is the hive's main storage in this video, which is slightly blurred, but you see a honeybee generating this wax scale from its just like it secretes it from its body. This is how they store it on their sort of back of their bodies, and then it chews it and very meticulously puts it like in a very specific position to build a small part of one honeycomb cell. So you can imagine the amount of effort that goes into building the full big structures that you saw. And there's, to understand how this is formed, we need to understand the cost of it, which is actually very expensive. 4 honeybees need to eat 8 pounds of honey to produce 1 pound of wax. So it's like super costly, and they it's amazing that they use this hexagonal structure that is mathematically proven for years that is the most efficient way of dividing a surface into regions with minimum material or walls, basically. But we do also know that bees don't always, not all of the structure, the comb is hexagonal. Bees have to deal with what we call geometric issues or puzzles. They have to build, sometimes feral bees build nests inside tree cavities around the branches. So the structure is not always like really even for these hexagons to appear. So this is kind of what gets us curious about if we can design experiments to deliberately test this. This is our apiary. This is me and the people who have helped me a lot throughout the years to collect this massive data set about the structures. These are commercial hives that we use, and these are filled with this kind of wooden structures that we call just like a frame. And if you just put it inside of this and come back in less than a day, this is what they built as their like part of their home. So we took what commercial beekeepers do is that they give the beehives these pre-made foundations with plastic hexagonal cells, so they can sort of give them a head start and get them to build faster and give them the honey faster. So they sort of build on top of. So this is, you can see in this is wax, and the plastic foundation is the bottom. So we thought we could use this. So instead of the pre-made plastic, we can use 3D printing to print our own puzzles for the bees. So this is what the things we tested are these three different categories. I'll start with shifts and tilts. I'll put them together. Basically, shifts is just very simply we sort of disrupt the distance between the given hexagons, so that one full hexagon of their choice of their desired choices does not fill, or one or two does not fill. So they have to sort of figure out. And then we tilt some part of the lattice compared to the other, so they can sort of just like be creative and think what to do in the. And they have to do this because at some point this is like a distributive process that groups of bees start building comb in different sections of the hive, and then they have to have a coherent structure for it to function. So the main question is basically what happens in that merging sections. So this is an example of like the type of lattices that we 3D print. The blanks in the middle, the gaps in the middle are just like for the bees to fill, however, and we didn't have any idea of like what it could look like. But after, like in this case, after 15 days, we can get a semi-complete frame. We can choose our part or the region of interest. We took pictures of them like regularly, so we know when. Yeah.
So this is all being done in the dark.
No, in the dark, you mean in the hive? Yeah.
So are they using tactile sensing to build a model of the physical world and the exact structure? We know this.
I mean, it's not known, but like
there's not vision.
There is vision, but it's not like our vision. It's probably dark, but the bees are able to see in dark. Is what is known. Like the spectrum of their vision is different from us. They can see in dark, but they cannot see like all the colors like we do.
I'm thinking more about the specific geometry of what they're trying to do.
But the geometry is visible to them. Yeah, totally. Yeah. Yeah, so we can take this region of interest and then apply just computer vision to detect the individual centers of the cells and boundaries of the cells, and this will be basically the basis of the results I'm showing you later. So from the pictures, we basically are able to reconstruct the structure, and we manually check it and make sure it matches and all of that. So we're pretty confident that this is what the bees have built. So in terms of the angle, you can see on the left-hand side, as we increase the angle of the tilt between the two sides of the lattice, they start using this adding this five-sided and seven-sided pentagons and heptagons pairs. And they do it in a very sort of systematic way, and in repeated experiments, we saw this happen. And they also sort of seem to have an idea of like how tilted the two sides of the lattice are and how necessary it is for them. So they really don't want to use any of the defects in the case when the two lattices happen to be exactly on the same side, with the same angle. They don't have any problem building hexagons on their own, which is what we knew already. What we didn't know was that these 5- and 7-sided cells have this specific nice pattern to them, and when you sort of like very roughly quantify it across for I think for each angle we have at least 10 different data points or full frames, I should say, and we get this sort of nice increase in the density of of the defects, we call them. And this is just a term we borrow from crystallography, because then later I'm going to sort of connect this to that field and to a lot of similar systems that show these defects. So if you see defects here, it's just a borrowed term.
I'm not totally understanding kind of the relationship to like what bees do naturally.
Yeah.
Right, because I think what you said is kind of this happens naturally. They'll
Yeah.
like start in different places and kind of
Yeah.
have to. So do you see kind of 7-sided and 5-sided
Yes.
in nature and all that stuff? Okay.
Yes, I'm going to show like this picture I showed you here. This one, we've analyzed it later after the fact, and you can see it there too. And a lot of other studies have shown it after this too. So, so then with the shifts, it looks like, I mean, it's less interesting, but it's also interesting in the sense that if you increase the gap, it doesn't hurt them. So as long as so they're able to fill the gap as long as it is with hexagons, but then in order to sort of connect it to the next lattice in the side that has like a specific angle of tilt in this section, in this example is 30 degrees, which is the maximum you can tilt. They use the same patterns as they used before. So it really looks like I don't know what this is, but like the comb orientation is basically the most important factor for the patterns of the defects they use, and not like if there is longer shifts or shorter. Yeah. Yeah.
How do you find density?
Yeah, so this is all in the paper, but so the density is basically how, so let me, I think the way we define it is the number of defects in a specific, for a specific scale. So, and the scale is roughly the number of rows, because there can be like different.
Less density.
Exactly.
Same density this
Exactly, yeah. The number of rows is what we look at, because if you look at the size, then bees sometimes build bigger or smaller cells, and that's not what we want to measure. We just want to measure at the level of the individual cells how the density changes. Questions? Yeah.
Yes, I'm very unfamiliar with this field. Is there like a sort of textbook understanding of how bees normally form hexagonal combs, and does that make any prediction about this?
Zero. No one knows how bees build cells. Is the million-dollar question maybe, which is why the model I'm going to show you is going to raise a lot of questions. It's not behavioral at all. The behavior is not.
Besides this one that you were entertaining might happen, like what's the most different from this outcome that could have happened that was plausible to you before you started the experiment?
I mean, honestly, I, as a graduate student, didn't have any idea. I don't think even my advisor had. But we honestly didn't think this like really ordered pattern would arise. What we thought, and what we sometimes see, just like when you're looking at frames of honeycomb, is that there are cells that just like don't look like the other ones. They look like this was an error. Or there was this other speaker yesterday who was talking about studying error. I really feel like I started thinking I'm going to study the error bars, but looks like the error bars are actually more organized and structured than we thought. So, I mean, it could have been just like, I don't know, random, just like globs of like things that will just like fill space and let the other, but no, it's like really sophisticated and organized. Yeah.
Now that kind of a soccer ball.
Yeah. Oh, that's, we tried to
have like different shapes, right? You don't feel hexagons. So
I
like wasps that build their nests around
exactly. Yeah.
You can find the pentagons inside.
Definitely. And then the numbers should be like a specific number for it to make sense. I can't tell you, so at least the time that I was there for 3 consecutive years, we tried to get the honeybees to build with different experimental setups to build comb on a sphere, but never with success. So, I mean, it's super hard experimental, but I would, yeah. Yeah, I'm sure they do, yeah. 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 Yeah, it's that's the thing is like before this has not been shown like quantitatively. So it's like we do have a sense of, I'll show you, so like in the picture I showed you, there are like three different structures, three different sort of, how do I say, groups of hexagons that are connected together with one line of defect of some sort, depending on the angle. So it's not, I don't know the number off the top. I mean, I don't think it's studied, but like I think after, like by the end of the talk, you should sort of have an idea. Like I have an idea, but I don't have a number, sorry. So I will just like maybe show this a little further. So we tested this pattern because when you look at the pattern, you might think this might be an artifact of the linear gap we gave them. So we played with the boundaries. We give we change the orientation. So like instead of putting them like vertically, we put them horizontal so that the gaps are horizontal. But the similar patterns appear. We also, this was like the hardest thing I did was in my PhD was to be able to extract the circular data, because it's just so important when is the right time for you to be there and take a picture of it, so you can get them before they're filled with honey, which is like not detectable and stuff like that. So, but like in general, with some error, it's very hard for the bees to build this. They abandon these frames until the end of the season. When they have to build something, they'll build something like this, but still we could trace the sort of similar tracks in there. And this is to your questions, I think. Once we knew this existed, then we went ahead and looked at the free comb, what we call it, without any puzzles, and we sort of spotted these similar patterns there too. So in a way, we can now do the opposite sort of prediction with this like very qualitative understanding of the structure of the comb, is that when we see this pattern, we know that the angle of the two lattices, what the angle of the two lattices might be, depending on the density and the structure of the pattern. So we really, just by looking at the qualitative data, experimental data, we really could argue that this wax is used in a honeycomb, in a honeybee group as a sort of a spatial memory or a physical memory that sort of stores the structure in itself. So if each individual changes the structure slightly, this will be remembered of some sort as for the whole group, and then the next person, the next bee who sees this change will sort of be able to modify its behavior based on that to get the coherent structure. So bees do not actually need, in this way of thinking about it, they do not actually need to use any sort of brain or any blueprint or any leader. They just need to sort of look locally and interact with the environment. So the environment or the wax is used here, I'm arguing, as an interface of some sort of embodied cognition, if you will. And the math of the lattice then will ensure that this structure will self-correct and will end up being the full thing that we see. So to sort of make sure,
I understand the logic of this. When you actually give the scaffold, that itself is the memory, right? You're talking about in the absence of a scaffold, the lay down wax is the memory, but here you're actually giving that scaffold, no?
I mean, we are giving the scaffolds for controlled experiments to test them, but we are seeing that happening in even without scaffold scenarios. Yeah. So we can then, I mean, they have to connect these two parts of the comb. It shows very clearly now that this part has been built with a different angle and this part with a different angle, and they sort of have to merge it. So we sort of are sort of transferring the logic from the controlled experiments and testing it on the real world data. So to make sure that we actually know what the math of the lattice is, we are modeling the final stage of the structure. This is, should be very careful to say that I'm not claiming that this is how bees built this structure, but the final structure, if you just want to figure out why it looks like that or why it's organized like that, this will be a way, I would argue, to get to that final structure. Just using, and this is again based on a lot of other systems that we studied, and we figured that they have similar patterns or of defects. A lot of them were using the sort of idea that the final structure is minimizing some sort of energy. And then using just like a slightly different or slightly changed Lennard-Jones potential energy, we're thinking about the final structure of every cell could be a trade-off between some sort of attraction and repulsion. In the sense of the bees, attraction could be with just their social grouping, they want to stay together, and also the odors of the wax, if you want to talk about the attraction between the individuals and the substrate. And the repulsions is just we can think of it the limits of their body. They cannot like really change one place at a time. Yeah.
They sit inside the cell and sort of rotate and deposit.
What, like how much the cell is built. In the first video I showed where the cell was not there at all, they start from the surrounding. And then that's actually a very good question. In the next section of the talk, I'll show you when you change the cell sizes, because the size of the cell is correlated with the size of the bee. So they can totally fit in it. One bee can fit completely in it. So yeah, if it's like half built, they can sort of go in and sit in there.
Before cells are constrained by their body size.
No, this is all after, like when it's completely done. Yeah. So you can assume, just this very high-level model assumes that just to match our experiments, we assume fixed cells that are the 3D printed ones, and then just some moving cells in the middle, which can be built anywhere. So the moving cells is only at each time step in this computational model, we'll move one random step with probability P and just will accept or reject the move if it leads to a minimized energy of the full lattice. And this is, through a simulated annealing process, we can just repeat this. And if you look at what the original structure looks like, it's pretty random. They're just like points that we throw in the middle. The number of the cells are data driven, so we drive that number from the data, but the positions are not. And then as the model sort of cools down, you can see that the structure is similar to what we get with the bees. And then to sort of make some sort of biological sense from it, we hypothesize that if this outcome is optimized or has some use, it should minimize the material that is used. So we look at some of the cell walls, and it's sort of showing a decline. The reason it's not like super high, the numbers, is because I'm just showing the sum of the cell walls of the full lattice, and most of the lattice is just hexagons. So if you like focus on the patterns, then you'll see a bigger difference. Now we want to sort of compare with the results from simulations. This is all experiments, and this is like on the side I'm showing the simulations, which are really very closely matching what we see in the experiments. So we can also sort of quantify the same factors, but I don't really know if the bees, I mean, the idea is that we could show with some sort of model that bees could use local noisy interactions. And the importance, I think, here is that noisy interactions are sort of built in our model, and it sort of at least calms me down a little bit that there is some sort of relevance, but otherwise, like the way there's no way to that I can convince myself that bees have an idea of like the energy minimization or optimization problem. Okay, so I'll quickly go to the last geometric frustration category that we check. We know that bees build different sizes of cells. So we 3D printed. So S is the size of the cells. This is the normal size. And this is like we push the limit of the size on both sides. So the smaller cells and a lot of larger cells. So if you are not convinced already that they're creative, I think you will be. So this is kind of the full pattern spectrum that we tested. If the cells are too small, they just avoid the whole thing. They fill all the gaps with wax and they start from scratch. They rather do their own thing than just the stupid thing we give them. If it's too large, then they again avoid it. They just build their own wax structure. But in the middle, a lot of amazing structures. So if it's a slightly smaller, and here I'm showing CT scan of the structure of the comb we gave from the top. So the comb is sort of growing as it's being built in different sections of the picture, and you can see that they are deliberately combining some of the cells to sort of make up for the. Yeah, exactly. And then if you look at the statistics, it's kind of about, and at least in our data set, it's about 24% of the cells are combined, which is like very close to the amount of cells that we sort of removed. And also the cell sizes, the distribution of cell sizes are very similar. It's definitely more error prone in the sense that it's not like strictly what they want, but they get very close to their desired size, no matter what the pattern look like. If you then there's this spectrum of the cell sizes that we from 1 to 2 that we tested with the bees. They're also able to sort of change their strategy in the sense now they're building tilted cells. So the tilted cells have the specific function. Even this normal honeycomb is tilted to some extent, but you can see as we increase the size, they increase the tilt as well. So with this scan data, we can compute all of those, and it's sort of a linear relationship.
Always the same solution.
Always the same solution for this range. Yeah. So from 1 to 2, and then for
a different colony.
Yeah, these are all from different colonies. These are all randomized. Yeah.
6 different directions.
The tilt is always in one direction. Yeah.
But what
just because, I mean, it's mechanical stability of the comb, right? These are filled with a lot of like very heavy material like honey or the brood, which is like super heavy. You cannot tilt it in all different directions. There is a left right symmetry. We didn't study it, but there is another group that studied, and they found exactly that. Yeah, we didn't. I mean, in this case, we were just like so busy with our question that we didn't look at it. But yeah, that's a great point. Yeah, there is symmetry. But then you would think that the three times larger would also be the same strategy, but they again changed their strategy to this sort of 2-layer comb now. So instead of just like tilting more, which would have been probably inefficient, they made this 2-layer structure that sort of very effectively, I think, transformed the huge or ineffective patterns that we give them to their desired, like very close to their desired size. So if you look at the distribution comparison between 1 and 3, you'll see that the cell sizes are now matching very well. Yeah.
So this kind of goes back to his question earlier, but if the building construction is inside of what it's building, then its body size determines effectively the size of the tube that it builds. That was happening, or
so not in this case, right? So this is twice the.
Like here, there's no existing structure that is body size. So
yeah.
Imagine it uses the sensors, the hairs on its body to
yeah.
determine whether it's the right size.
Yeah.
So it's going to build a foundation, and then it'll start building something of the appropriate size
yeah.
by spiraling around.
Yeah, that's generally true. Yeah.
So wouldn't that explain almost everything here? Like that they're basically building from within each of these cells, and therefore it's going to be its own body size.
But then it doesn't explain this one, because this is twice. The cells are twice bigger, and it's also not only the what.
They don't use them.
They do, yeah.
These are the-
They do all the, so whatever I'm showing you has a function. And this is like in the paper, and I can describe like specific functions to you. Nothing that they build is unused. Everything is usable. Yeah, sorry. Like, no, I don't. Yeah, yeah, yeah. I mean, I'm almost done. Yeah, this was my summary. So all these other systems have similar structures in them, and this is what we use in the modeling.